Auzubillahiminash-shaytanir-rajeem. Bismillahir-rahmanir-rahim. Dear students of 12th class, subject Botany, Boys Higher Secondary School, Nagri. Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, in our yesterday's lecture, we have discussed the post-fertilization events in angiosperms, and we have come to know that different types of developments take place after the fertilization, and which are called the post-fertilization events in angiosperms. And among those developments, one development was the development of endosperm, and other developments are development of seed, development of embryo, etc. And among all those developments, or we can say in other words, all those post-fertilization events, we will take the development of endosperm. So our today's topic is development of endosperm. In angiosperms, endosperm develops from triploid primary endosperm nucleus by triple fusion or vegetative fertilization, and it is the food-laden tissue formed during the development of angiosperm seed, which provides essential nutrients to the growing embryo and young seedling at the time of seed germination. Dear students, as you have understood in our last lecture yesterday, that in flowering plants, or we can say in angiosperms, the endosperm develops from triploid primary endosperm nucleus by triple fusion. Triple fusion here means by double fertilization. Double fertilization is also called triple fusion or vegetative fertilization. So in angiosperms, endosperm develops from triploid primary endosperm nucleus by this triple fusion or double fertilization. And what is this endosperm? It is nothing but a food-laden tissue which is formed during the development of angiosperm seed. And this endosperm, it provides the nutrients to the growing embryo and young seedling at the time of seed germination. Now, based on the first and subsequent divisions of triploid primary endosperm nucleus, the endosperm is of three types. This means that after the first and subsequent divisions of triploid primary endosperm nucleus, endosperm is of three types. It means that the triploid primary endosperm nucleus undergoes different types of divisions, one after the another, and it forms the endosperm. And this endosperm is of three types. One is called the cellular endosperm, another is called first one is called the nuclear endosperm, second one is called the cellular endosperm, while as third one is called the halobial endosperm. First of all, we will discuss the first type of endosperm, that is the nuclear endosperm. This nuclear endosperm is most common type of endosperm and occurs in mustard, sunflower, and coconut, etc. So you should remember that this nuclear type of endosperm is formed in the plants like the mustard, जिसको हम कश्मीरी में तिलगुल बोलते हैं, and the sunflower को गुल आफ्ताब and coconut को खोफर. Nuclear endosperm it is a common type of nuclear endosperm in these three types of plants like mustard, sunflower, and coconut. So what happens during this endosperm formation? Here the primary endosperm nucleus divides repeatedly. The primary endosperm nucleus it divides repeatedly. It divides The divisions go under it undergoes divisions one after the another by pre-mitotic nuclear divisions. What type of divisions occur in this primary endosperm nucleus? Repeatedly, it undergoes the pre-mitotic nuclear divisions and it forms a large number of free nuclei in the center of embryo. एम्ब्रियो के सेंटर में बहुत सारे फ्री न्यूक्लियाई बनते हैं. एम्ब्रियो सैक के सेंटर में बहुत टाइप के न्यूक्लियाई बनते हैं विदाउट द फॉर्मेशन ऑफ वॉल्स, और उनके वॉल्स नहीं होते हैं. Then a big central vacuole appears in the center of embryo sac. Then after these divisions, a central vacuole appears in the center of embryo sac, which pushes all these nuclei formed to the peripheral cytoplasm. तो ये साइटोपेरिफेरल यानी कि साइटोप्लाज्म के एक तरफ इन तमाम न्यूक्लियाई को ये पुश करता है. कौन? ये central vacuole. Thus, then the free nuclei come to lie between the embryo sac. फिर ये फ्री न्यूक्लियाई पेरिफेरल पोजीशन के बाद ये आते हैं और एम्ब्रियो सैक के बीच में बैठते हैं. Then cell wall and vacuolar membrane and the wall formation occurs, and after that wall formation occurs. Finally, whole or most of the embryo sac is converted into multicellular endosperm. और इस तरीके से lastly embryo sac is converted into multicellular endosperm which then encloses embryo. और ये multicellular endosperm embryo को enclose करते हैं, बीच में रखते हैं और ये इसके इर्द गिर्द होते हैं. तो this was the nuclear type of endosperm formation. So this was all about this today's lecture, development of endosperm, and today we have discussed the first type of this endosperm, that is the nuclear endosperm. In our tomorrow's lecture we will study the second type of this endosperm, that is a cellular endosperm. Now I will show you diagrammatically the development of endosperm or the development of nuclear endosperm. So this is the diagrammatic representation of nuclear endosperm formation. You see first in figure A, zygote is at peripheral position and centrally is located primary endosperm nucleus. Then in the second diagram, this how the central vacuole occurs and how the this free nuclei are pushed towards the peripheral position of the embryo sac. This is in second diagram, nuclei and endosperm. Then in the diagram C, in figure C, you see the multicellular, this type of all these nuclei enclose the embryo and it is shown by diagrammatically. So these are the different stages in development of nuclear endosperm. It is a diagrammatic representation which we have studied today in our topic. So this was all about today's lecture. Stay home, stay safe. Thank you very much.